Hello, everyone. Good afternoon. My name is Alexandra Bridgeland, and I'm with Millipore Sigma, specifically at the Rockville site. And today I will be presenting on a novel bioinformatic approach for strain-specific virus detection using NGS. So we received a client request to be able to differentiate the 6 different strains of avian leukosis virus using NGS, being able to differentiate them and also to detect them at low levels in a host sample. And so of note, 5 of the different strains are pathogenic, while one of them, strain E, is non-pathogenic. So we wanted to be able to at least differentiate all of the pathogenic strains from the non-pathogenic strain. And part of the motivation behind this project was that ALV can cause cancer in chickens. And so this might be useful to potential stakeholders in chicken health, specifically in the agricultural industry. And so one challenge we had was that the references for ALV were not always available or reliable. So we needed to use some creative methods to overcome that roadblock. So our first purpose was to create reference sequences for STUs. And so we created 6 different consensus sequences for the 6 different strains of ALV. And then our second purpose was to develop a method in which we could have a host sample or a sample from a spiking study, and then use this method to be able to detect ALV and also differentiate by strain. So this slide gives a big overview of the methods used, and so I'm going to break this down in the upcoming slides. So our first step was consensus generation, our first version. And so we had raw sequencing data separated by strains. So we had sequencing data for each strain, and we wanted to use a pre-existing in-house variant calling algorithm that we have. This algorithm uses alignment and pileup to create a consensus sequence. The only problem is that we need a reference sequence as input because it does do alignment. And so we selected these reference sequences based on the completeness of the sequence and also if it had a paper alongside it. So then what we did next was we wanted to validate these consensus sequence using de novo methods. And so we took the same pre-processed sequencing data, and then we aligned them to the version 1 consensus sequences generated from the previous slide. And then from there, we extracted the aligned reads. And the reason we did this was because there was lots of background in the sequencing data. So we wanted to remove that background and then use those reads for the de novo assembly. So this table on the slide depicts the number of contigs that were created through our de novo assembly. So you can see that it's a couple hundred for each of the different strains. We wanted our contig lengths to be between 7,000 and 9,000 base pairs because that was the length of our consensus sequences for each of the different strains. And so you can see when we filter for contigs over 1,000 and contigs over 5,000, that there were a decent number of contigs. And so then our next step was we took the contigs from each of the strains and mapped them back to our consensus sequence just to validate some of these contigs. And so this was one example from strain A specifically. And you can see here that this was sort of an ideal case. So you can see that the contigs, there's multiple contigs that span the whole reference length. However, this case for strain D was a little bit more fragmented. So you don't have a contig that spans the whole reference length, but however, between the various contigs that we did get, they do span the consensus length. So our next step was we filtered these contigs to only include those greater than 1,000 base pairs, separated by strain still. And then we blasted against ALV sequences that we downloaded from NCBI. And we curated a list of the highly similar sequences. And then from there, we clustered at 95% to create a curated list that we then used in another iteration of consensus generation. And so basically, we employed the same method that we did for the first version of consensus generation. However, this time we used the new reference sequences created from the blasting. And so from there, our results seemed a little bit more in line what we wanted. Just there was a little bit more reads that mapped to these new reference sequences. And then also the consensus sequences were a little bit longer and a little bit closer to the expected length. So then we compiled all these results into a 37 sequence final database. And so this database consisted of 11 contigs, 20 NCBI reference sequences, and 6 consensus sequences. And the 11 contigs, we narrowed it down to 11 contigs based on clustering. So we clustered at 99%. And then we also did some manual curation. So for example, if there are 2 sequences that were very similar, but one was longer, then we would take the longer one. And so we were able to narrow it down to 11 contigs. And then we used the 20 NCBI reference sequences that were from the curated list from the BLAST results. And then we also used the 6 consensus sequences generated. So then that leads us to our proposed experimental procedure. And so what we proposed was we would take raw sequencing data, and the sequencing data would basically be from either a sample from a chicken or from a spiked sample from the lab. And we would use our 37 sequence database and then use the same variant calling algorithm. And then we would have a table of our variant calling results. And it's a little bit misleading to say variant calling results. It's more so alignment results that come with our variant calling algorithm. So this is an example of what the results might look like. And so we conducted a small study and we wanted to validate this procedure. So basically, we took just sequence data of the host, and then we subsampled reads from strain A, and then added these into the host reads to at 5%. And then from there, we looked to see, we ran it through our workflow to see what the results might look like. And we were able to come up with this criteria for evaluating the potential hits. So we looked at the percent of reads mapped and also the consensus length. So for the percent of reads mapped, we looked at greater than 0.01%. And then we looked at consensus length greater than 1,000. And so I highlighted the 2 greatest hits, and you can see from the reference sequence column that they do match with ALVA, which was what was expected because that is what we spiked in. And then the other potential hits are likely just due to homology. Then in addition to this, we also made a database of unique regions, and we also made a database of unique SNPs. And so basically, we were able to create this database through multiple sequence alignment. So we aligned all the consensus sequences and compared them. And then also, once we were able to have a list of potential regions and potential SNPs, we looked at all of the sequences related to a single strain. So for example, we would then perform a multiple sequence alignment for just strain A and look to see what number of regions and SNPs are in that specific strain. And displayed on the slide is an example of what this database might look like. And so you can see displayed, you have the unique regions, and you also have the positions in which these regions occur. And that is all I have to share today. So in conclusion, we were able to create a 37 sequence database for viral detection, and we were also able to develop a method in which we would use this database to detect ALV at low levels in chicken cells. And we also developed a SNP database as well as a database of unique regions for ALV that are used as strain signatures. And our next steps would be spiking studies. So instead of performing the in silico spiking studies that we performed, it would be experimental spiking studies. And this would just experimentally validate our results. And so just some acknowledgements. So thank you to Brad and Rebecca, who are the client-facing individuals on our team and helped guide this project. And then thank you to my specific team. So Yanfei is the expert on our team, so she was our leader throughout. And then thank you to Hero and Will, who are my colleagues and helped develop these methods. And if you have any additional questions for me, I put my email on the slide, so feel free to reach out. Thank you so much.
Time for a couple of questions. So I have one.
Is this broadly applicable? I mean, could you do the same thing broadly over a whole range of different kind of viral?
Yeah, you could apply this to whenever you need to do viral differentiation.
So just reflecting off of Caitlin's talk before you, is there a need for the contigs? Could you extract features out of the reads without to detect strains without going through assembly?
We did the assembly as further confirmation. So I think if you didn't want that confirmation, then I think you could just use the reads. But yeah, we wanted to further confirm our results. And we did cluster the contigs to our consensus sequences, and we noticed some small differences. And so that's why we kept them in the database.
Any other questions?